Hey guys, my name is Juliana Peterson. I am a human biology pre-med major from South Dakota State University where I'm a junior. I'm originally from Westbrook, Minnesota. I'm going to talk a little bit today about my research that I completed on campus in the summer of 2018. I completed research with Dr. Jeffrey Clapper. He's a reproductive physiologist in the Department of Animal Science at SDSU. A little bit of background, my project was on evaluating the anterior pituitary gland leptin receptor expression in pregnant versus non-pregnant gilts. So successful reproduction in swine is dependent on the correlated synthesis and release of many hormones. At the level of the hypothalamus, we have GnRH, which is gonadotropin releasing hormone. Specifically, I looked at the expression of the receptor that hormone binds to. That's going to go and stimulate the synthesis and the release of luteinizing hormone at the level of the anterior pituitary gland. Luteinizing hormone, we looked at the specific subunit of the beta, so we say LH beta in that instance for graphs, and then we looked at leptin. Leptin is a hormone that's secreted by adipose cells or fat cells. Previously, it has been loosely correlated with having involvement of regulation with LH and LH beta, but not a strong correlation, so we wanted to investigate into that. For reference here, we have a quarter, which is next to an anterior pituitary gland and a posterior pituitary gland of a pig. That anterior pituitary gland, or that U-shaped one, was where I was extracting most of my RNA, my mRNA specifically. So we had 24 crossbred gilts that had their estrous cycles synchronized, and then they were artificially inseminated. 30 days after that insemination, we slaughtered them and collected blood, anterior pituitary glands, and as well as their reproductive tracts to determine pregnancy. From there, their anterior pituitary glands were bisected mid-sagittally and snap frozen and then stored at negative 80 degrees Celsius so that we could preserve that mRNA that was hanging out in them. From there, we did a total RNA isolation with that 1/2 of each of the anterior pituitary glands from each of the pigs. They were treated with DNase and then reverse transcribed through PCR, so we used reverse transcriptase to look at 3 specific things. So the porcine leptin receptor, mRNA, so this receptor that leptin hormone is going to dock onto, the GnRH receptor mRNA, and then the LH beta mRNA. We produced cDNA from there, and then we used random hexamer primers that are listed in the table 1 here on your right side of the screen. We used those primers to grab onto the mRNA, and that was our ability and how we were able to quantify the amount of mRNA for each of the 3 previously listed hormones and receptors. So first things first is our first hormone that I'm going to talk about is LH beta. This is the most important one because it was the only one that was statistically significant. That's why we have an asterisk on it. So we can see that the pregnant's on the left and the non-pregnant's on the right, and there's an asterisk above the pregnant. So it was actually downregulated by 0.1 or 0.813 fold in the pregnant versus the non-pregnant. So that was interesting and statistically significant. Moving on to our GnRH receptor, so that receptor that hormone will dock onto, you can see the levels vary, but they're not statistically significant enough to make us want to dive into that research more or do further experiments, wondering why the levels aren't the same in the 2 groups of the pregnant versus the non-pregnant. Most importantly for my poster and my project is we have our pregnant up here and our non-pregnant over here for our leptin. So this is our leptin receptor mRNA expression. It did not differ between the pregnant and non-pregnant gilts, which was interesting. So that's kind of the conclusion. It really did not make a difference. We did see a difference in that LH beta. However, what we were looking for was leptin to change, and it did not in our experiment that we ran. So further experiments should include other considerations. One of them should maybe be animal adiposity, so the body weight or the body type of the animals coming in, so that since leptin is secreted by those fat cells, that wouldn't be another variable that could maybe sway why or why not there's no difference in that leptin hormone receptor concentration. And having that said, really no difference that we found in the leptin hormone differing between pregnant and non-pregnant gilts in this experiment that I completed with Dr. Jeffrey Clapper.